So, hello everyone, and welcome to the second day of the 16S workshop. This is going to be a quick presentation about other tools, tutorials and methods available for QIIME 2 and 16S analysis in general. So we're going to talk about some of the QIIME 2 online tutorials that are available, some other tools and methods, some other genes that you can use for metagenetic analysis, and I'm also going to talk about PyCrust 2. So, QIIME 2 tutorials and guides. So there's many guides and tutorials for QIIME 2. You can go onto this link and it'll help you give practice and show how certain analyses can be carried out. So definitely one of my suggestions is when you're learning a new process, if you can do multiple tutorials, that's always useful. When I first learned 16S analysis, I went to a workshop and then I did an online tutorial, followed by another online tutorial, and then I started doing analysis once I felt more comfortable doing it. I was thankfully given the time to do this. I know sometimes time is an issue, but I will say that you'll probably save a lot of time in the long run if you do some tutorials until you're comfortable with it before trying to do your own analysis, if you haven't done much before. And also, so what we would call commands, QIIME 2 calls plugins. So if you're interested in all the various commands QIIME 2 has, you can go to their plugins page. So let's talk about some of the tutorials, give you a brief intro to them. So the first main one they have, their kind of introductory tutorial, is the moving pictures tutorial. It's an online tutorial. You can download all the data, it'll have all the commands. And it is analyzing the human microbiome data. So we talked about this a bit yesterday with the 2 individuals, the 4 body sites and 5 time points, and looking at kind of antibiotic usage and how it affects these individuals. They also have a fecal microbiota transplant tutorial, so where they're looking at children with autism and gastrointestinal disorders. And the idea here is, if you give them a fecal transplant, will it improve their gastrointestinal disorder, and will this then in turn improve their autism? Will it make life better for the children, and make it so that their autism isn't as severe for them or the people around them, I guess. And so that'll be interesting, even if it doesn't directly contribute to autism. It still might improve their quality of life in general, and therefore obviously it's quite difficult. And then there's the Atacama soil microbiome tutorial. So this is looking at the Atacama Desert in northern Chile, which is one of the most arid locations on Earth, with some areas receiving less than 1 millimeter of rain per decade, so incredibly dry. And it's looking at the soils profiled at 2 east-west transects, being Baquedano and Yungay. So that one could be interesting if you're interested in extremophilic bacteria and their environments. We also have a Parkinson's mouse tutorial. This is looking at fecal samples from humanized mice, trying to determine the relationship between Parkinson's disease and the fecal microbiome. And so they got humans which had Parkinson's disease and transplanted their feces into predisposed and resistant mice, and seeing how that affected the mice, whether they got Parkinson's disease or not. So as well, QIIME 2 now have their own YouTube channel, so the developers may have made a YouTube channel. I think they made this last year, and they've been producing more and more content with it, so that's probably a good resource to look at as well. So we've been talking about QIIME 2, but there are other tools out there. So 4 of the main tools are QIIME 2, MapSeq, Mother, and QIIME 1. In days gone by, people either used Mother or QIIME 1, which were pretty similar to each other, used a lot of the same algorithms and biodiversity measures. And people either used one or the other based on what they were first taught. There were a lot of people who would go out and say, oh, this one's definitely better than the other one. So there were QIIME diehards and Mother diehards. Personally, I was under the opinion that they were both pretty good, and generally to go with the one that you learned how to use I was taught Mother, so that's what I used. But then once QIIME 2 came out, I then jumped to that because of the ASV methods, whilst QIIME 1 and Mother use OTU clustering. The following paper recommends using QIIME 2 or MapSeq. I personally would say go for QIIME 2 because that's what I'm teaching you here, so it's probably you have a better understanding of it than MapSeq. There's also a lot more resources available for QIIME 2. A lot more people use QIIME 2 than MapSeq, so it's probably generally an easier tool to use in the long run anyway. So some visualization. So QIIME 2 has some visualization techniques and tools. They're pretty good, but they can be a bit limited, so if you're interested in other things. There's Megan 6. I've not used it myself, but it is a microbiome analysis tool for metagenome, metatranscriptome, or amplicon data, so not only for 16S data, but other applications too. The one I would use outside of QIIME 2 is phyloseq, which is an R package to visualize metagenetic data or community data in general. It doesn't have to just be 16S data, but it's generally what it's used for. However, it does require R and ggplot2 fluency. So you'll need to know R and you'll need to know ggplot2 before you go into phyloseq. Now, some people might say there's a bit of an issue with phyloseq is that it seems to be abandoned by the developer. They don't seem to be working on it anymore. However, it's still very popular, and there are new packages that kind of use the phyloseq language to continue on and develop and make more tools. So even though phyloseq is maybe not being developed anymore, there's a package called microbiome that builds on top of phyloseq, so that's introducing new things, and so I think still makes phyloseq very viable. And there's more information about the R packages in the supplemental book down, so if you're interested in phyloseq and microbiome package, then you can look at it there. And then there's another tool called VAMPS that some people seem to like. I've not used it, but it is a publicly available website to visualize microbial population structures. But I'm pretty good with R, so that's why I go with phyloseq instead. So phylogenetic tree visualization. So part of the step of what we're doing in our QIIME 2 analysis is we're making a phylogenetic tree. There is a way to export this phylogenetic tree into a NUIC file, which is the general all-purpose tree file format, and that's in the supplemental book down. And so you could then view this if you want. There's various tools. There's iTall, which is the interactive tree of life, which is a nice web tool to quickly view trees. There's Figtree, again another tool. There's Archaeopteryx, and there's many more. Now, it's always a question of which phylogenetic tree tool should I use. And this link down here, this BioStars forum post, somebody asked which tree tool should I use, and somebody gave a very good answer as to listing a bunch of different phylogenetic trees visualization tools and when and why you should use them. It pretty much comes down to the size of your tree, so the number of branches your tree will have. Small trees, certain tools are better for. Medium-sized trees, certain tools are better for. There's large trees, and then there's very large trees. I think very large trees were greater than 1,000 branches, and I think he mentioned Archaeopteryx is good for that. And so yeah, I think it's a very good post to I have this saved in multiple places, so I can refer back to it if I'm going to be visualizing a tree. And then we have metagenetic biomarker detection. So we talked a bit about this on Tuesday, looking at biomarkers. We used ANCOM, and NICE is also available in QIIME 2. However, there are other tools such as LEFSI, Maslin, HALA, and DESeq2 if you're using phyloseq. You'll notice that these first three are all from the Huttenhauer group. They're a pretty well-known group when it comes to bacterial community analysis. And these tools can all be used for shock and metagenomic data as well. Okay, and then there is also metagenome functional prediction through metagenetics. So there's various tools. I'm gonna go a bit more of a deep dive in this, but some of the tools are PyCrust, phylogenetic investigation of communities by reconstruction of unobserved states. So you can see they're really like trying to have nice anagrams by making really long, complicated names. There's tax for fun and there's pie filling. But let's go back on to pie crust in a wee second. But first, a brief mention about fungal metagenetics. So we talked about 18S sequences briefly, where you can use the 18S sequence instead of the 16S if you're looking at eukaryotes. But if you're specifically looking at funguses, you might want to use the ITS, the internal transcribed spacer. There's two different regions, ITS1 and ITS2. It's found between the 16S rRNA and the 23S rRNA. And various tools you can use to analyze it are CHIME2, Pipettes, and Clover ITS. None of the tools are perfect, and I would definitely recommend checking your classifications at the end of it. With that, let's go into PyCrust2. So PyCrust2, as I said, stands for Phylogenetic Investigation of Communities by Reconstruction of Unobserved States, version 2. It predicts, with heavy emphasis on predicts, functional abundances based on marker genes. So it'll try and predict the KEGG orthologs, the enzyme classification, and the metasite pathways that are within your samples, your real samples, based on the taxonomy information that's in your samples. So based on the taxonomy table. And it does this with greater than 40,000 full 16S sequences, both from our bacteria and archaea, from the Integrated Microbial Genome Database. But how does it do this? So let's go through its pipeline. So it has quite a long pipeline. There's more information about this in the supplemental book down. So if this is something you're interested in doing, it'll show you how to carry out the pipeline in there using data from your 16S analysis. As you can see, it has 5 different steps. Step 1, align ASVs to reference sequences. Step 2, place ASVs into the reference tree. Step 3, infer gene family copy numbers of ASVs. Step 4, determine gene family abundance per sample. So we had it for ASV, now we're going to get it per sample. And then step 5, infer pathway abundances. So let's go a bit more in detail of step by step. So the first step, we're going to align our ASVs. So this is our ASVs that we have produced through CHIME2. So kind of our representative sequences we get from the data to denoising. And we're going to align these to our reference sequences. So we have the greater than 40,000 reference sequences in our database. So this is kind of an example of how you do that. So we've got our ASVs, 1, 2, 3, and 4 in this case. And let's say instead of, was it 40,000, let's say we have 4. We have 4 reference sequences, A, B, C, and D. So we align these. So we can see that ASV 1 and 4 align to A, ASV 2 aligns to B, ASV 3 aligns to C, and none of them aligned to D. So now we have our aligned ASVs. And with our aligned ASVs, we also have a reference tree. So as well as having the sequences of the references, we also already have them in a tree. And so with our aligned ASVs, we're going to place them into the reference tree to get a tree of ASVs and reference sequences. So let's say this is our initial reference tree that's already in the database. And with our alignment information, we're going to place our ASVs into our reference tree. So we can see here that we have a clade of A and B that have now ASVs 1, 4, and 2 in there. We have a separate clade of C and 3 now, so reference C, ASV 3, and then reference D is all by itself because there's nothing aligned with that. So aligned our ASVs, placed our ASVs into a reference tree, and now we have in our database our gene family copy numbers per reference genome, and with this information in our reference tree with now our ASVs in it, we're going to infer family copy numbers of ASVs to get predicted gene content per ASV. So let's say we have this table, we have gene families 1, 2, 3, 4, 5, 6, and 7. We have the abundances that these are present within each of our reference sequences. So reference genome A has 10 of gene family 1, abundance of 2 for gene family 2, abundance of 7 for gene family 3, etc. And then we infer our gene family abundances per ASV, that are ASVs, based on kind of the similarity of our ASVs to the reference genomes. So now we have our nice inferred gene family copy numbers of ASVs. So before I continue, does anyone have any questions on any of these steps? Okay, grand. So now we have our predicted gene content per ASV. However, this is only so useful. Instead, we want to determine the gene family abundance per sample. So we want to know how much of each gene family is in each sample based on the ASV abundances and predicted gene content per ASV. So this is an important note. It's a slightly controversial thing. Some people, so PyCrust2 developers think you should do it, but nobody else does this. And whether you should or not is, I think the consensus, generally speaking, is to not do this. But when we analyze 16S data, we are naively saying that every single 16S is one individual. So each 16S sequence we're kind of saying is one individual and came from only one individual. But the way biology actually works is that certain bacteria, so certain species, will have more copy numbers than other species. So one species might have one copy of the gene in its chromosome. Another species might have 2 copies of the gene in a chromosome. Another copy might have 4, etc., etc., or 3. So let's say we have, these are our samples. These are the number of 16S sequences per species. We've got our 3 species. We've got the green star, blue triangle, and orange cube. So here in sample 1, we'd initially say there are an abundance of 4 for the green stars, 4 for the blue triangle, and 4 for the orange cube. However, these are the 16S copy numbers for each of these species. So what PyCrust2 does is it normalizes the number of each ASV's abundance based on its 16S copy number that it has in the database. So in this case, we would go 4 16S sequences divided by its copy number of 4. We therefore only have 1 in our sample. 4 blue triangles divided by 1 is 4. And then 4 orange cubes divided by its 16S copy number of 2 is 2. And so that's for one sample. We then do it for our pink sample as well. 8 divided by 4 is 2. 3 divided by 1 is 3. And 4 divided by 2 is 2. So this is only PyCrust does this, saying because of biological reasons. Does anybody have any questions about this, or did that make sense? Okay, grand. Oh, does someone have a... Okay, no worries. Great. So it does this first. It then has our sample abundances, so our samples S1, S2, S3, S4, let's say, and our ASVs of ASV 1, 2, and 3, 4. These numbers have been normalized by the 16S copy number. We combine this, our ASV table, by our gene family abundance per ASV table. And now we have a gene family abundance by sample table. So you can see we've got sample 1 has a gene family, has an abundance of 400 for GF1. Sample 2 has an abundance of 200 for GF1. Sample 3, 270 for GF1, etc. So now we can have a comparison of the gene family abundances between the different samples. And these gene family abundances being contributed by every single organism within that based on the 16S data. So now we've got a predicted sample gene family profiles, but we also then want to map these to the gene families. We want to map the gene families to the pathways, sorry. So the pathways being KEGG, the enzyme classifications or EC numbers, and metapsych. And so we now have our inferred pathway abundances. And so then you kind of have pathway information. However, I've probably been like scaring you away a bit from it because I kind of hesitant to say that these are true results. And there's some key limitations. So this link has some more key limitations and information on it, but let's go through some here. So one of the key limitations it has is the sequence placement in the reference tree. So let's say we had our reference tree and our inserted ASV sequences in here, and this is the way it went. However, once you start introducing more ASV sequences or different ones, it can dramatically change the way the tree looks. This might be an extreme example, but let's say we add in ASVs 1, 7, and 6. Maybe these are from some more samples, so we've discovered these in a different way. Now we can see that ASVs 4 and 2 have actually moved off to 4 being with D because 7 was here, and so it kind of changed the way the alignment weapon happened. And then 2 has gone with the C clade because we introduced a new ASV 6. And so if you've ever like messed around with phylogenetic trees where you've got like maybe 10 sequences, you make a tree, you see what it looks like, and then maybe you add in another sequence and you see what that looks like, you will notice that the clading will change dramatically. It's not a simple matter of that new sequence being added in somewhere, it's the whole tree being reorganized because we have like further information. So a tree of 10 different organisms will look very different than a tree of 20 different organisms with those same 10 organisms in there, most likely. So this is one limitation. So depending on the data you input, you might get very different results. Another limitation is the availability of reference genomes. So 40,000 reference genomes sounds like a lot, but in the grand scheme of all bacteria and archaea, it's not that much, especially when you're wanting functional information of everything. So here's an example using birds. There's a lot of information out there about the reference, like the information on duck genomes. So let's say your data had lots of ducks, you would be fine. But let's say your data had kiwis, we don't have as much information out there. Its genome is not as well, it's not very well characterized. So therefore, you're probably not going to get very good information. Maybe you have something like lyrebirds instead, and their genomes are not characterized at all. So if you have organisms with no information about them, you're going to get very poor, if not, and you maybe will get no information about it. And then let's say you have organisms that are unknown, you're going to get pretty much no information about it. If it's close to something that we do know, that's fine, but if phylogenetically speaking it's not close, then unfortunately you're not going to get any information about that. And this is a picture of a thunderbird, which is a mythical animal in this case to represent the unknown. You have environmental biases, so this kind of ties into the availability of reference genomes. If you're working with human microbiome data, you're probably going to get relatively good data. It's very well characterized, lots of people study it. If you're working with animals such as rumen, cows, you're still okay characterized, but definitely not as good as the human microbiome, so you're going to not get as much information. And let's say you're looking at like the microbiome of plants, such as like the root microbiome, it's going to be very poorly characterized, relatively speaking, compared to humans and compared to cows. So then just briefly talk about some of the other things. So I kind of touched on this, but if you have ASVs that are not similar enough to a reference, i.e. they don't align well enough, they can't be added into the tree, these ASVs will be excluded, so you'll be missing information about your data. And so that's where I said if you have organisms that aren't known and aren't close enough to anything else that we do know in the database, then it'll be excluded and you won't get any information. You can only predict genes represented by Kegworth logs and EC numbers. So again, you might be interested in a specific gene, but if it's not in the database, you're not going to get any information about it. The pathway mapping is based on mapping of the KEG or EC to pathways, which may be wrong. So this information is really useful, the pathway mapping information, but it's not foolproof. There's a lot of information in there, and probably not all of it is correct, unfortunately. And one thing to note as well is that your predicted functional metagenome is biased by the 16S sequencing. So we talked about the biases that come in from Illumina sequencing, on Tuesday, but then there's also biases that you're adding in through the analysis. So you're kind of like compounding biases on top of biases, and then this PyCrust2 is biased itself. So biases from the Illumina sequencing, biases from the analysis, and then from the QIIME2 analysis, and then biases from the PyCrust2 analysis. So there's a lot of issues. So at the end of the day, you have to be very careful about how you talk about any information you get from PyCrust2. It's very biased. It's not the actual picture. It's all based on predictions and inferences. So it's more kind of like a hypothesis finding tool. Like you have use it like to discover some hypotheses, and then try and back up these hypotheses based on maybe some lab work or doing shock and metagenomics. So it's kind of, yeah, get as much information as you can, but again, it's not very, very reliable information. And so that's the end. So I'll stop recording.